So good afternoon and welcome to my talk on abundance estimation and differential testing on strain level in metagenomics data. That was not the plan. Okay, so metagenomic deals with the analysis of genomic material which is directly taken from environmental samples. So here we consider a complex mix of bacteria and we want to ask who is in the sample and how much of them. And in this work we want to specifically focus on strain and substrain resolution. And in the next step we compare metagenomic samples and we ask which of the strains change their abundance and which of them will hold a constant abundance. And for this comparison, it will be also important to know how accurate the abundance estimates actually are. So it is our second goal to also capture the abundance estimation variance. And I also want to note here that this variance is not to mix up with technical or biological replicate variances. We're going to apply next generation sequencing. So we receive millions of reads which we map back to reference sequences in order to identify the origin of the read. But on strain level, we will observe many highly similar reference sequences. So we face the problem that a read may map to different genome sequences equally well. So if we conduct a normal mapping approach and we simply count the number of read hits per reference genome, then this might be a mapping abundance result which we may observe on strain level. We have here in blue the true abundance proportions for the taxa in the set, and in gray we see the mapping counts we observe. And due to the shared reads, we will frequently observe on strain level abundance overestimations and also abundance calls to actually absent taxa. So in order to achieve a high quantification accuracy on strain level, it is very important to tackle a fine resolution of shared read counts. So I want to emphasize that this problem here is really about read count resolution, and we do not necessarily need to know the exact origin of the read. So let me just summarize shortly our objectives. So we want to focus on strain and substrain level here, and so we aim to achieve a fine resolution of the shared read counts due to the highly similar genome sequences present. It will be important to also capture the abundance estimation uncertainties, which we also want to integrate for further differential testing. So here I present our method HiTSeq, which consists of the three main parts of mapping, taxa abundance estimation, and differential assessment. And as a first step, we recommend to do a pre-filtering of the reference genome sequences by using fast and external profiling tools. So in order to pre-filter the data set for species expected in the data, and here to really specifically focus on strain resolution. Then in the mapping step, we map all the reads to the selected reference genome set by using pseudo-alignment approaches. Pseudo-aligners here are sufficient and also fast enough as we do not require an exact alignment. We then count the number of mapped reads per reference genome and save it in a vector to which we refer as the mapping abundance. And importantly, so here we include also all the multiple assignments of the shared reads. And then next, we construct a matrix in which reflects all the similarities of the reference sequences. So the matrix holds all reference sequence comparisons, and one entry of the matrix refers to the probability that a random read, which is derived from genome I, can be potentially mapped to genome Y. We then use this similarity matrix in a model in order to resolve the shared read count bias which we observed in this mapping abundance vector. And we follow up on this core idea by our former tool GASIC, but now we present a new generalized linear model which is based on absolute counts, and the GLM is defined by an identity link function. And further, we also introduce a Poisson error term to capture the abundance estimation uncertainties. And this equation system will then be solved by an iteratively reweighted least squares. As a result, we will obtain corrected taxa abundance estimates along with standard errors which reflect us the estimation accuracy. So next, if we want to compare the abundance of a taxon between samples, we first of all have also to account for the potentially different numbers of input reads of the samples. So we need to apply normalization factors to make the samples comparable. And then we will use the abundance error, and we will build an empirical abundance distribution for each taxon. And we will do so by doing a two-step sampling process, which is based on Poisson distributions. So basically, instead of comparing abundance point estimates of a taxon to compare between samples, we will compare entire abundance distributions. And so the idea is that the divergence of these distributions then reflects the differential abundance. So basically, if two abundance distributions are clearly separated from each other, then this will point to a significant abundance change, while an increasing overlap, of these two distributions will mean less change or even no abundance change. And we measure this overlap of the abundances by creating a difference distribution, and from this difference distribution we also infer empirical p-values. As a final result of the whole workflow, we receive an output table with a list of all the taxa and all the reference sequences we put in, along with abundance estimates and log two-fold changes and statistical p-values for our significance assessment. So now I'd like to show you the application of our method to 3 different data sets here. We used the frequently used benchmark data set I100 by Mende et al. Further, we used sets from a recent Metagenomics challenge, the CAMI challenge, and we also have simulation sets in which we created a higher density of strain clusters. So we considered different number of reference sequences as input, and also a varying degree of reference similarities and different densities of strain classes in these data sets. So the density of strain classes is basically shown by the number of species in the set in relation to the references we are considering. And here to the right, we have an example of a similarity matrix of our simulation set, which shows us the reference sequence similarities which are present. We see in the bottom left corner here a large E. coli cluster, which also holds different substrain clusters of very high sequence similarities, like greater than 97%, and also along the diagonal, different other strain clusters having high sequence similarities. Further, we also consider different number of input reads and different read characteristics, and also a very different abundance complexity profiles. So the I100 dataset was frequently used for tool evaluation, and just so in a publication just recently, Schäfer et al. Showed that the new RNA-seq tool Callisto also provides excellent performance in metagenomics abundance estimation, and there's quite a few abundance tools out there, but not many really tackle the resolution beyond species level. And Callisto was shown to have a superior performance concerning strain resolution in comparison to in comparison to existing methods like Bracken, Clark, Garsig, and Express, by now showing smallest average error and smallest relative root mean square error values. But we also have to note here actually that Clark and Bracken, they do only provide resolution on higher taxonomic levels. So it was very nice. We could reproduce this result table from Schäfer et al., and we could even further show that DTASIC achieves even smaller abundance error values. We can see here the high accuracy of DTASIC in this plot. So in the red line refers to the true abundance proportions of the taxa, and the blue dots refer to the abundance estimates we received. And one of the main reasons for the improved performance is that DTASIC also allows an accurate resolution on substrain level. So from this data set here, we can look at an E. coli cluster which holds 2 substrains which share a sequence similarity of around 97%. And in this bar plot here in yellow, we show the true abundance proportions in yellow, and we use the blue color to overlay the abundance estimates of the tools. So here to the left, we can see that Callisto struggles with significant overestimation and underestimation for these substrains, while we receive a better resolution with DTASIC here. And we observe similar results also in the Kami set. So here we have an example of a Corynebacterium cluster in which DTASIC achieves an almost perfect resolution within the cluster, as can be seen by a perfect overlay of the colors. And in comparison here, Callisto assigns almost equal abundances to the 2 substrains, which again here share a very, very high sequence similarity. And also overall here we can see again small abundance error values. We switch now to evaluate by using the sum of squared error values instead of, as you saw before, the average error and the relative root mean square error values, as the sum of squared error values also allow to penalize absent taxa calls, and so this measure makes it a bit more precise. So on a side note, we did not run Garsig on this set as it takes a very long runtime. We further used simulation data to also investigate the impact of absent strains on the method. This is also it's a very crucial point because, of course, it is very hard to define an optimal reference sequence set up front. But we could show here that DTASIC correctly identifies absent taxa in the set, and that it also ensures an unbiased abundance estimation of strain clusters which hold both present and absent strains within one cluster. The method actually also allows to distinguish here down to substrain level, as can be seen by these marked taxa in the plot, which, so it distinguishes the absent and the present ones, which form together one strain cluster. And again here we could reach smallest abundance error values. So finally I want to also show some results on differential assessment. Krami provided 2 condition sets with different abundance complexity, and we compared these samples with each other, and we can see that DTASIC achieves a very high fold change accuracy, as can be seen by the plots along the diagonal. We compared to a tool called STAMP, which is not doing the shared read count resolution, is also not integrating abundance errors, and we observe less overall accuracy. And further, we considered the computed p-values based on our statistical framework, and we see that they can enable a clear separation between differentially abundant and non-differentially abundant taxa. We also spiked in some non-differential events here with a fold change of 0, and we can see that they all receive p-values greater than 0.05. And all taxa from the Kami challenge who showed clear fold changes above 0, they all received p-values close to 0. So to sum it up, HiTSeq represents a new approach for taxa abundance estimation and differential testing, which also allows resolution down to substrain level. Therefore, we introduce a new generalized linear model for shared read count resolution, and we also integrate the abundance uncertainties. And HiTSeq shows its particular strength in the resolution of substrain clusters of high sequence similarity. And yeah, I want to also refer to our webpage, where you can find a HiTSeq manual with all individual steps, which is like shows you easy guidance through all the different steps too. Yeah, and also the code is available from there. So here's some references, and also for more detailed reading, I want to refer to our recent publication. So finally, I want to thank my co-authors, Bernhard Renard and Benjamin Strauch, and the whole group of bioinformatics from the RKA, also the DFG and the ISMB ECCB for the funding and giving me the opportunities to speak here. And thank you for listening, and I'm happy to take questions.
Yeah, we have time for 1 or 2 questions.
Thanks for the great talk. I was just wondering, in terms of the running time, and I'm sorry if I missed that, so you did compare to Callisto, which is known to be very fast.
Yes.
So I imagine that if you do mapping-based, it's quite time-consuming.
So for the actual pseudo-alignment, we use actually part of the Callisto framework. So we use the Callisto pseudo-aligner for the mapping. So not its resolution, like not its shared read count resolution, which they use an EM framework for doing this. So we take apart from this and just take the pseudo-aligner. So basically what concerns the mapping, we will have the same runtime. And what is, of course, a bit cost expensive for us is the construction of the similarity matrix. So it is important for us that we have a pre-filtering of reference sequences up front. But then the matrix construction will just scale linearly, basically. And yeah, the thing is also, if you consider this for different samples on the different conditions, you just have to calculate this matrix once. And so, for example, for the Kami set with like 230 reference sequences, the whole run took maybe around 5 to 6 hours, the whole analysis.
Cool, thank you.
Yeah.
So you had 3 data sets, basically 2 real data sets and a fake data set. And you used the fake data set to figure out how your algorithm performed when species were lacking. Why didn't you just make fake reference genomes instead and choose a normal data set?
Well, I mean, one of the main rationales also for these data sets here was that we wanted to compare to recent publications as well, and also to tools who benchmarked on the same data sets to make it also more comparable, and not coming up with a completely new data set where we have not as much reference to before, but it's for sure a good idea, and yeah, should be done in the next rounds, I would say. So thank you for this comment.
Okay, in the interest of time, we are going to take the other questions offline. Our next speaker is Andre Kales.